down for the fish, and they lay down rings every year like a tree almost. And we're able to sample these ear bones, the otoliths from the fish, take them back into the lab, and under a microscope, you can count the rings on these ear bones and tell how old a fish is. There's some cool branchy business there, Abby. Branchy sponges.
Is that another cod? Cow cod up on the...
It's just out of my view.
Yeah.
But we do tend to see a lot of them down here in Footprint. And there's more of the Acanthoborgia.
Rockfish book says the vermilion max age is at least 60. This die. And Bob is wondering about the origin of the name Footprint. I don't know, Bob. If you look, it does look like the outer step of a foot, but I actually don't know what it is. It's right next to another bank we call Piggy Bank.
That's
I don't know
clever.
who the first one to name that was either. Somebody with a good sense of humor.
All right, I'm going to change this to 115 for one move.
Guy is crediting Milton with the name Piggy Bank. She says it does resemble a pig. There's something about funding in the story. I would love to hear Milton tell it. So Milton Love is the rockfish guru. Wrote literally the book on rockfish and has done a million dives on this very reef in the submersible back in the day, along with folks from the Southwest Fisheries Science Center. So for those tuning in now, we're at 220 meters south, 220 meters depth, just south of Anacapa and Santa Cruz, the channel that goes between Anacapa Island and Santa Cruz Island in the Channel Islands National Marine Sanctuary in Southern California. We're moving along a ledge, collecting species specimens of corals or snails or sponges as if we encounter something we think is new or we're not quite sure what it is, to help us ID the biodiversity of the sanctuary. We've also been conducting quantitative transects, counting fishes and corals and sponges and figuring out where they are in the sanctuary and what abundance, how common or rare they might be, to help us better manage this protected environment. Leslie and Julie asking any amazing discoveries, the salad bowl sponge and also the shortbread sea star.
Is shortbread sea
We've
star
been-
an official name?
Very, very, very food oriented this trip.
And it's for
We're-
posterity, Abby. No.
The 4 to 8 shift is very food motivated.
We're all food motivated, I think.
Yeah, it's not
Just a heads up, guys, video is going to go ahead and switch out, so Al's coming in.
Raj,
Okay.
thanks, Marley.
Yeah, thanks for a good watch.
Thanks, Marley. Oh no. I think Meredith has it with the Acanthogorgia. Thanks, Dodai. We're looking for some more of those round, flat sponges. Kind of pancakey looking. Maybe grab a bit of one of those. We just finished a transect in a nice sponge area. We just collected one of these bowl-shaped sponges, the white funnel that the lasers are coming through now, to try to determine what it is.
Science, did we hear back about if they wanted this coral kind of coming up, the yellow one?
I think the current sample we have is from this area, so we may be holding off to try to find it somewhere else.
Roger
within
that.
the sanctuary.
Sure
Thank
thing.
you.
Yeah. ROV switching out.
Thanks, Jess.
Yeah, thanks you guys. Thanks, Jess. That
Yeah,
was
thanks
great.
you guys.
Urchin pile. What's going on?
Wow.
Why is she excited?
Well, let's get to that.
That deserves a Zoom.
Yeah, we need to
Seems to be an
get a better
urgent
look at
meeting in progress.
Wah, wah, wah.
that. Is that just like a kelp holdfast or something that tumbled down? What are they?
It does look like that, doesn't it?
All right, Nav switching out. Thanks, guys.
Good night, Ben.
Good night, Ben. Thanks.
So, Lizzie, I'll sign out and let Chris give you the lowdown if you're ready.
Hey, thanks, Erica. It was great to do the watch with you. I appreciate all your wisdom.
Yes, thank you. It was great to learn about the sanctuary.
You bet.
And Abby, are you looking at that branching sponge beneath the urchins? Good night, all. Thanks.
What are they chewing on? Talk about food motivated. All right, Lizzie and Julie, are you on?
Yes, we are.
Oh no,
Hello.
I was
Welcome,
not muted for
guys.
that. Drinking my straw.
Getting your dinner? Look at that. Can we get a zoom on that pile before we leave the area? And just
I
see
know,
what
I'm
they're
really
chewing
interested in that too.
on. As I focus in on that, so we went along, Lizzie, as we had kind of suggested, but there was really nothing happening until we got to that steep area. So I guess some of this has probably been traversed already by
Yeah.
Southwest Science Center because we're going right along the slope. But it was, we went above it and there wasn't much happening. So we've kind of just
It's a
stayed
kelp
along this,
holdfast.
following the same contour. Yeah, that's what I thought.
Yeah.
They're totally loving it.
That is like, you know, just the bonanza in the deep sea when that comes down. Look at that. The urchins are happy tonight.
So you said you tried to go up on the lip, Chris,
Yeah,
and there wasn't much?
I think it's been mostly interesting just hanging out at this sort of, just following the same contour along.
Mhm.
That's kind of where we found anything. A little low, at least where we were, tended to get pretty barren again, and up higher tended to get barren again pretty quick. So my suggestion is to kind of continue on like this along the contour over to your next waypoint, Z2.
Yeah.
And then at any point, if you're finding cool stuff and it takes longer, at any point we can always bail, go north to the ridge, and do our habitat mosaic,
Okay.
or just head up to Z4. So we don't have to get all the way to Z3, I guess.
Yeah. Yeah, maybe we'll keep going and see. I almost want to. Oh, yeah, that's Z2. Let me see.
So how do these urchins know where that is?
Oh,
like
yes.
they came
Okay, thank you.
Far and wide.
Yeah, Shoreside, let me know if you want
Smelled
me
it.
to zoom or in or out on HYPACK as you figure out waypoints.
No, that's great, Sam. I like what you said, Chris. I think we're going to keep going in the same direction, kind of following this ridge face, like the actual slope part, in the direction of Z2. And after we get past Z2, if it gets boring, we can go straight up to the ridge, or we can kind of follow along to Z3.
Like in a line.
Because I'm also anticipating the watch following us and
might be
leaving
helping
them
him
plenty
far.
of things to explore.
Is
Because we'll have
mine out?
about 7.5 hours of bottom time left.
Hey, Chris.
Yeah, they did say they're going to be off at 700. Go ahead, sorry.
Oh, I'm sorry. Did you guys find more of the sponge species or the images? You know, she sent about 5 images of different sponges she was interested in collecting samples of. Did you guys find more of them?
Coming in for the kill.
We collected, like if you look at the top of your screen, that kind of, I think that might be it. Maybe not. It's another base sponge. We collected what we've been terming the salad bowl sponge. Which
The one that's sort of
looks
open,
like fancy, yeah.
like a bowl. Okay.
Yeah. That's been our main successful sponge collection. We collected a coral right at the beginning and finished up with some shell hash for Daniel, and we did 2 transects during our watch. The last one we pulled a Niskin, I think we pulled the Niskin bottle at the end. So there's 2 Niskins left. I think there's 2 or 3 push cores if you see a good spot for that. Yeah.
Cool. Okay.
All right. Well, I will sign off, at least off the audio, but I will continue listening to you all. I hope you have fun.
Thanks, Chris.
Thanks, Chris. Enjoy your evening.
Yeah, thank you.
Alrighty, well, Shoreside, I caught most of that. Sounds like we're headed towards Z2, continuing along this contour line. Anything you want to do here before moving on?
Also 45 are needed. Okay. No, I think we can keep going. I haven't seen anything in the science chat other than Daniel was saying there was a brachiopod, but I don't think we're zoomed on that area. Daniel, let me know if you see a brachiopod that you want to collect. Otherwise, I think we can keep moving along the slope face in the direction of Z2.
Roger, and so brachiopod here or continue on to find a brachiopod?
Daniel is saying keep moving.
Keep moving.
We'll continue.
Roger.
So in looking at those sponge images, image number 1, I thought I saw a little bit of it. It's hard to tell if it's like a broken salad bowl looking sponge or if it's a different one. So anyway, we'll keep our eyes open. And we did collect the 4th image, which is that sort of barrel sponge with the clean outside surface, but there's another one, the last image looks like a sort of a oblong sponge that has a darker surface on the outside of it. I haven't seen any of those. Have you? I mean, we'll have to look for it and see what we find, but. Okay, so Abby's saying that they're not sure if this is the broken salad bowl, if it's the same sponge species. And I think they've seen the last one too, but they just haven't sampled. A zoom will help. So we'll keep the search going for those sponges.
While we're getting set up, shall we do a round of introductions since we just did a shift change?
Sure.
I can start it off. My name is Lizzie Duncan, and I work for the Channel Islands National Marine Sanctuary as a research ecologist, and I help coordinate some of the deep sea coral work that goes on the West Coast, and that's why I've been lucky enough to be chosen as one of the chief scientists for this expedition, this leg of the expedition.
And I'm Julie Bursack, and I'm a colleague of Lizzie's at Channel Islands National Marine Sanctuary. I'm the team lead for education and outreach. And yeah, I'm excited to plug back into the expedition tonight and continue this. This is a long dive we've been doing today. Started at 4 a.m. and it's continuing through tomorrow, correct?
Yeah,
Yes.
we did shorten it by one watch to make room for a little bit of a longer of a dive after this because it was originally planned to be 32 hours. So now it'll be 28.
Oh, okay. Hey, Lizzie, really quick, look at that sponge up there. It's in the view right now.
Mm-hmm.
And
Above the
I'm gonna,
lasers,
I think
like kind of tucked in.
above the lasers, yeah. And then it might resemble sponge specimen number one, the number one image. And I think Abby is still on the science chat. And she's
Yeah,
saying,
she is.
please.
Zoom, zoom.
Please zoom in. So let's see what she says. Got a lot of little squat lobsters around it.
Yeah, lots of little anemones.
Okay, so she's
Abby's
thinking this
saying-
one does
Oh,
look
go ahead.
similar. The texture is similar to that of the salad bowls. So we can go ahead
But
and
if
zoom
we
out on that
could
one.
take a
Hey, zoom
snip
that
to
out.
confirm.
Oh,
Yeah, we could-
okay, yeah.
What's that?
Yeah.
What are you doing?
She would like a snip of it to confirm that it's not,
BridgeNet.
it might be different. So good, let's take a snip of it.
Full position.
So is this a slurp or where's this going?
She says a snip.
Are we just snipping it to snip it, or are we putting it somewhere?
Is it a snip and slurp or a snip and box?
Snip and
Let me
release.
translate.
If it's a small snip, I would slurp it. If it's a big snip, maybe the box.
Okay.
I mean, you know, it's a
Well, if it's a floater, then the box is the other question.
yeah, that wouldn't work so well.
Well, it depends on. If you put it up front, it's okay. If you put it in the side, then it's more of a problem.
We've got two sponges forward already, so forward box is out of the question.
Okay. And then Daniel also would like us to slurp some of the white clam brachiopods to the right of the salad bowl sponge, maybe 3 specimens. So let's work on the snip first, then we'll look at that.
Got it.
I have a little issue with the claw here.
What was the species that Daniel wanted, Julie?
The clam brachiopods to the right. Yeah.
that enough? How much you want?
It was just a little, I think that's good. Yes, that's good. Oh, it's loose.
I think we're gonna get the whole thing.
You gonna get the whole thing?
I just feel like it's gonna come along, but you should try for the snip.
Yeah, you can see the small rock that
Perfect.
it's on in
All right, jar 2, or
Yeah, jar
Speed up.
2's ready.
Is that enough? Too wimpy. It's pretty small. The deadly silence of silence of science.
Can you say that 5 times?
Well, I can't see the snip. It's so small. It's hidden by the
small.
claw.
All right, full blast on jar 2.
Okay, ready?
Hopefully it goes in.
Get in there.
Uh-oh.
It's there. It's got it. Oh, is it gonna make
Too
it?
rigid for
It's.
slurping?
It's not that small.
Uh-oh.
it's going.
Stuff it in there.
Yeah, but... Oh.
How many
Which
beans
jar is
are
it
you
going
giving
into?
it?
Yeah, can we add sound effects to the telestrator?
It's going in 3.
Oh, it made
it's
it.
in 3.
Oh,
No,
2.
two.
Sorry.
2.
Fascinating.
2.
All right, it's in there.
Is that enough? Let off the juice? The bean?
I
Yeah.
think Abby says that she thinks
No
it's
beans.
okay. So now let's look for the brachiopods that Daniel's interested in. They were just to the right of the sponge. So let's zoom out. Yeah, there we go. Zoom out.
Do I want to flush between samples here, Steve? Given that we have...
Are we
No, I
sucking
mean,
brachiopods?
we didn't slurp up any dirt, so you don't need to flush. I think we can move on.
That's just clam brachiopods. Yeah.
Can we zoom in?
He's spotted some to the right of the sponge. So we need to kind of zoom in and see what we find.
Like in the dirt over here?
I haven't noticed them yet.
They're not all of these shells that we're looking at, are they?
Oh, there's
On
a lot of
rock.
shells.
He's saying here on rock.
The right shape for brachiopods, but I thought their shells were a little rounder.
Oh, is there a kind of like more towards the upper left? It looks kind of.
Yeah, he says those little things.
Oh, the little things. No, I
How
think
little?
we're talking about the shells, right? All the shells that we're seeing are
Yeah.
actually brachiopods that are living.
On the rock or?
And on the sand?
Yeah, oh yeah, I
On the
see it.
side?
See the yellow sponge area there?
Oh, yeah,
Just
yeah.
above
Oh.
the yellow sponge. Yeah, yeah.
Right. So all those things are then?
Looks like it.
Yeah, all the white brachiopods.
So you see there's a there's a
there's-
brachiopod right above that yellow sponge.
But
Oh.
there's one that's open and
Yeah.
confirmed to be an actual
Yep.
animal, where these might be shells.
Okay, is this
Yes,
a slurp?
and Daniel's confirmed. I don't know.
I think
Looks
it's
pretty small.
yeah.
Yeah.
Yeah, brachiopods usually have that is attached by a muscular peduncle versus like a calcium carbonate type of attachment. So
Steve, the telestrator's on channel 3, or for everyone to know.
Yeah.
Telestrator on 3.
That's where we're looking.
Okay, so zoom out. So it's a slurp?
oh, the telestrator, that's amazing.
You're gonna slurp the peduncles?
Yeah,
Maybe a scrape
scrape and slurp.
Scrape and slurp.
Yeah, scrape and slurp.
Okay, it's pretty steep slope to
Scraping.
yeah.
Well,
Don't even try.
We can scrape with a hose.
can we
Just say it.
can we see if any of them are on the smaller rocks to the right of the arm?
To the
Yeah, what
right?
if we just
I
collected
can't
one of those
beyond
little
there.
rocks?
Maybe the left one.
Beyond my capabilities at this
And, like, are any in the sediment
point.
here living?
You're saying a whole rock to grab a rock?
Grab a rock, or if there are any that are looser in the sediment. I don't know if they're still living though.
Good.
Or
Zoom
pick,
in.
yeah, or pick up a small rock if we see one attached. So I think we would need to zoom back in a little bit and pan to the right to see what we see.
There's
I'm
not
gonna-
much pan.
Yeah,
I think that's it.
That's it's all
I
she's
was looking at
Okay.
the
got over there. I'd have to reposition.
I mean, that's a kind of small rock towards that little one right in front of in the sand.
I don't know if
I don't know. I can't tell if it's attached
that
or if it has the
this
right
little rock
critters
is
on
not
it.
the right stuff, right? Zoom in some more now. Are those? I mean, those are them, aren't they?
It looks like it. Let's see.
I don't know if any of them are
God, they
living.
look like. That's what Daniel. Several small candidate rocks. Any okay? Daniel, does this rock that we're seeing in the zoom right now look like there are living brachiopods attached?
Oh,
Oh,
what is
no,
closed?
Yeah. I would
Just one though,
say
but
that's confirmed.
but I wonder if the other ones are just the bottom
Yes.
shell.
Yes.
This rock would be good.
Well,
This is a good
Okay.
rock.
Yeah.
Rocks
Let's
are
see
good,
if we can
'cause then
pick it
we
up.
can put that in the starboard box easily.
Yep.
Okay,
Yeah.
zoom out now.
And so for those of you that are listening in, Daniel Geiger's with the Santa Barbara Museum of Natural History, and they're interested in brachiopods and some of the shell debris and for their collections at the museum.
The arms definitely need some work here.
It might
I hope
be
it's
attached.
I know. I'm
Like a
hoping this is still
protrusion?
pebble or a
I don't know. Doesn't
rock
really
and
look
not
like it.
part of the reef.
Gotta give it some more force.
Oh, that's a wide. It's getting ready.
Stretching.
Going in.
Yeah, warming up.
Can you?
Yeah,
Yeah,
it's not
pan there.
moving.
Pan?
It's not moving.
Yeah.
It's not even like jiggling a little bit.
Any other rock will do too, but yeah,
Okay.
if we can't get this one. So right now we are at
Ah.
223 meters. Nope, it's not budging.
Doesn't seem very budgy.
I got one on the right-hand side that's a good candidate.
Yeah.
Yeah,
More
that's
than
not
meets
even
the eye
moving
there.
a tiny bit. So yeah, it's okay.
So any of
The rockfish looking
these
in, checking it
pretty
out.
good.
Oh, I see,
So,
Steve.
Yeah, the
Yeah,
problem
there's
over
like
there
a
is I
little line
can't
of
zoom
them.
in on it. I just have to grab it unless I reposition.
Yeah, there's that one with the 3 in a row. Like an old straight
In a
line.
row, right here.
A little bit
We-
That's
right
what I
there.
was thinking, but like any of them, yeah.
We can do a little carousel rotisserie, see what it's got on it before we put it away.
Yeah, that one's loose. That helps.
Oh yeah, that looks like cobble.
Jesus, rockfish, sorry.
So curious.
Very nicely done. That took finesse.
Mhm.
All right, I'm gonna zoom in and see what we got. Maybe not.
Do we suspect any of those are living?
Let's see. Daniel, looks good to Daniel.
Yeah.
Maybe the top
Are they living?
one is at least living, I think.
Oh, that's a good turn on it. Yeah, let's look from this angle.
Oh, that's so cool.
I know. So it looks like the middle one is the one that's maybe living.
This is the jewelry case mode.
It's funny because they're like concave, so it almost looks like just the bottom half of the shell and the top half is gone or something.
That's what it looks. The
But
top
maybe
one looks like
that's
it's missing. It's been eaten. Like it's just the bottom part of the shell still stuck to the rock.
Do you want to grab another one just in case?
Yeah. Toss
After
this
this
one?
watch leads?
sure.
Toss
Yeah,
this one?
He
let's
says it
take
looks
this one.
good.
It does?
Let's take this sample.
Oh, he's the expert. Okay. And where's this guy going?
We can go in A. Small.
Is there floaties in the box?
There are no floaties that I'm aware of, but best to open it slowly.
Okay. Well, do we need to kill a thruster? That's
Probably a good idea.
okay. I might drift off of here though. But you want to get ready to kill the thruster?
How do I kill the thruster?
You got to go to the relay page.
Oh, yeah.
SV.
Relay. Oh, yeah.
If you only want to open it halfway, you can also go in F, which is where the scoop is right now.
Well, we'll just if we kill the thruster, we won't get the downwash. Just
The
still have
SV.
the. It's off.
Are you ready for it off?
Yeah. Yeah.
Okay, SV command off. Oh. Button didn't. Button. Oh, no. Yeah, there it went.
Get down.
Ready for it open?
Oh, I gotta get stable here. Yeah. Okay. All right, box open.
Going for F?
Wait, what was it? A you wanted?
A is ideal, F is okay.
A is right here.
Yep. Looks good. That's a sample.
All right.
Steve, this is sample 124 and 125, correct?
SV
Correct.
command back on.
Yeah. I get the slope here.
All right, back on.
Okay.
Science, anything else here that we'd like to collect or zoom on?
I don't think so. I'll wait to see if Daniel wants to request another rock. Otherwise, if he does not, we can continue on.
I think you just said that's all. Thanks for that collection. Oh wait, it just says that. Thanks for that collection. Danny, do you want any more samples, or should we move on? He's good. He has enough.
That will
yeah.
be fine.
All right, continue on our merry way. Bridge nav.
Very merry.
Thank you. So we have to continue on with our introductions.
30 meters.
Who's on the
bearing
Nautilus
100.
tonight?
I'll start. Samantha Wishnak, navigator.
And Bob Waters, I'm the Herc pilot.
Paul Glick, I'm the Argus pilot.
Al Santos, I'm the video engineer.
And on data, my name is Steve Oskovich, and I'm data logging and science managing for this leg.
Excellent.
Anchovy.
So Lizzie, those sponges there that have the growth on the outside, could that maybe resemble what Abby's looking for or not? There goes another anchovy.
Team from earlier. I rewound the Nautilus live video to see that. That was insane. I have never seen that many like swarm in a bait ball in front of the ROV. Sorry, Julia. I had to say that.
No, that's okay.
Anchovy, because that is just, it blew my mind, and it's worth everyone who has not seen it. Oh shoot. It's probably 12 hours from now though, so it might not be there anymore. But
we do have a video from Channel Islands a couple years ago that's a sardine bait ball.
Couldn't see.
Yeah, that was incredible. So
Yeah.
if you're on the Nautilus Live website and you search for bait ball, you'll find our video. There were also sea lions chasing after the bait ball, which was exciting.
Yeah.
Oh, that's awesome.
Actually, now that I think about it, sea lions I think were chasing after larger fish that were going after the bait ball, so it was a nice little food chain there.
Yeah.
Speaking of swarms, by the way,
Totally.
in Argus Cam there's some squid.
Ooh.
Market squid again. Ship moves underway.
Roger.
So Julie, what were you saying about the sponge?
Oh, I was just wondering with the, the sponge, that last sponge, picture, image number 5 that Abby sent, but it doesn't, it looks more oblong than round, but it's white. The pigmentation of the sponge on the inside looks white, and then it's sort of got a lot of growth or darkness on the outside. I'm just we're seeing some barrel-looking sponges as we're in this area, but they're more round. I don't know if the shape has anything to do with it, but that was another one that she wanted a sample of.
I'm wondering that too, because she calls those dirty barrel sponges, and we've passed some very, very, very tall dirty barrel sponges on our last shift. So yeah, I maybe we'll keep an eye out for short, chubby dirty barrels. Maybe that's a good thing to keep an eye out for.
The common name is dirty barrel sponge.
Like this one?
It's a loose descriptor. It's a loose descriptor. We're on the fly here.
But fuzzy dirty.
This is an opportunity for folks to describe what they're seeing in those sponges.
Because no, I think she refers this one. The dirty part is very long, and I know she calls something a shaggy barrel sponge. So this one might be the shaggy one.
Yeah, these look shaggy.
Oh, okay.
What's this orange one?
So really, she's just trying to find things or find phrases to call all of these so she can stay consistent amongst all her observations of these relatively similar groups. That way when she finally does get an ID, she can go back and essentially control F replace
You give me some leash.
with dirty barrel sponge, and then she can put it in the proper scientific name. So she's just trying to keep a consistent naming scheme.
Yeah. I mean, those are so those are the shaggy dirty barrel sponges.
Yeah.
Okay.
Is that yellow coral of any interest?
That is Acanthogorgia, and I feel like they have just passed one up.
Great.
So I will look at the collections list.
This is an interesting area right here. There's a lot to see.
Okay, well, I gotta get moving.
Gotta go.
Gotta go.
Okay.
Gotta go.
All right.
Unless
Okay,
we want to stop. Yeah.
Zoom out.
Sonar sure looks a lot better. Alrighty. Whoop. Yeah, it looks like this will fall along the slope for all the way to Z2.
So this is interesting. On this slope, there's a lot of sediment, but there's some little rocky outcroppings, and you can kind of see in the distance as we approach where the rocky outcroppings are, because you'll see the sponges attached, which is a good clue that we're hitting some hard substrate coming up.
Nice basket star.
Yes. And is that another salad bowl? That's a nice looking salad bowl there. That sponge.
Zoom in out. Oops.
For an image, that would be great.
Sorry.
Oh.
Dirt cloud.
Yes.
Oh, benthic siphonophore was zipping now in the top left corner.
Yeah, they were looking at one earlier on the dive. There's tons of them in this area.
Yeah, kind of
So
surprised.
similar depth to where were we the other day? Petrali had a lot of
Okay, you
those
can
too.
zoom in again now.
That's beautiful. So Abby's still on the science chat, so we'll see if there's this. I mean, I think we've collected samples of this, but we'll just confirm it.
You want to kill the laser?
And we're having some dialogue about describing dirty barrel sponges too.
I might have gotten wrong what she refers to as a dirty barrel.
Uh-oh,
Zoom
okay.
out, Al.
There are just so many variations of dirty sponges that we've seen.
Uh-huh. Yeah, pretty much.
So the ship has completed a move, so if we do want to take a look around this rock more, got time.
Okay.
You want to zoom in and look at it? What are you doing? It's got the same yellow coral in there, same place. Oh.
Hello, squat lobster.
You give me some more leash again?
I think you got leash, but
It says 22.
yeah, but there's still.
I don't know. There's a decorator crab. Zoom in.
Oh,
Hey,
cool
no,
master
Nope.
star. Oh, and a little crab.
Dirty
Oh,
carapace
it's so
crab.
cute.
Mm-hmm. Dirty carapace.
Different costume from last night, though. They don't have access to those zoanthids.
Yeah.
Yeah.
Oh.
What's it on?
This
Is that
one,
a sponge?
it's a sponge that Vasca
Yeah.
star's on.
Are we full wide on Argus?
So now I know all those shells around us are all brachiopods of some sort, which
Getting
Daniel's
a lot
going to
of
try to
yanking
describe.
about here. I don't
The
know.
tether's like loose. I mean, I know I'm high up, but it's not taut.
Getting in some current here? Does look like might be headed
I don't know.
across your brow.
Yeah, I guess. It's not me doing that.
It's not Hurricane, it's not Argus.
Mm-mm.
What's
I'm
the
not
third?
making the stick do that.
I think the
wobbling
ocean is
around.
the third variable.
Like an old Scooby-Doo cartoon.
Yeah.
Hey, Dana, can you go over the last 3 samples with me just so I can confirm what I have in my notes?
The last 3 samples?
Yeah, just want to
Yep.
make sure I have them right.
So we have on number 123, which was taken on the last watch, we have Niskin 3,
Okay,
which was taken at 219 meters at the end of transect 10.
got that. Okay.
124 was number 2 slurp jar, the white platy, possibly salad bowl sponge fragment. And 125 was the rock with several clams or brachiopods, and there was a sponge anemone on it as well. And that brings us to 126 is the next sample.
Got it. Okay, perfect. Thank you. That was very helpful. Oh, okay. So yeah, the shaggy sponge that I was trying to wonder if one of these that we were looking at stemmed from Erica Frew when the Nautilus was in the Olympic coast. They had seen a sponge with the really long spicules on the outside that caught a lot of the muck in the water, and Erica had called it a shaggy sheepdog sponge. So I was wondering if that was similar, but there's, as Abby was saying, there's so many variations on one theme, so many different types of dirty spicules. It's hard to come up with unique nicknames
getting
until you can get an identification.
Seems better now. I don't know.
So you think the current's picking up?
No, I just there's just some yanking on the tether there that was likely the current.
I mean, this
You see the football
this
floating
area is
around
really good for current.
Yeah.
Or just off frame,
Yeah, I
there's a
guess.
giant squid pulling on Argus.
Yeah.
Yeah.
Pulling on the
Well, it
long
is October. That
As
would
long
be
as it's
really
not
good for
fishing
this
line,
one.
it's
Ah,
Yeah, I know. Wow.
okay. Zoom out now.
alrighty, continue on.
Yes.
Another
Yep, please.
salad bowl there.
Yes, so I think Abigail is interested in sort of seeing if there are differences in the species of these different glass sponges, the salad bowl versus the barrel. So that's why we're looking at, if it seems like they're visibly different, collecting some of the a snippet of them so that we can do some genetic comparison.
So they're your buddies up there.
Oh yeah, we've been seeing a lot of benthic siphonophores. I don't remember ever seeing this many.
Gonna grab
This
bubble.
frequency.
Who's your buddy?
Benthic siphonophores.
Oh. That's good.
As one does.
You always get,
Zoom
good
in
to have a
out.
buddy.
Gotta find friends somewhere.
When you're out
That's
here
right.
for as long as us, anything's a friend.
They're just so cool
Just
and
need that
unlikely.
human connection, human siphonophore connection.
Human siphonophore. I mean, the fact that there's, a little colony happening here of parts all working together. It's like us. This is the control van. There's the swimming part and the stinging part and the attaching part.
So we're a
Can I do the
siphonophore.
eating part?
Yeah.
I think Sam
do
has
the
had
eating
more
part
social
first.
interaction with benthic siphonophores than with humans this year.
Hey,
Can you guys
buddy.
remind me what we're looking at here? Is this your siphon? This is it. This is your buddy?
Yeah, this is benthic siphonophore. Pineapple siphonophore is what they call them too. Look at
Oh,
how
I like
incredible.
that.
All the little opening and closing of the little filter feeder bits
Oh
at
my
the top.
gosh.
Wow.
Oh,
It's
they're
active
going
right
to
now. Or is
town.
that the Hercules effect
Wow.
on that, or is that? It seems it's like
It's going to town.
it is.
Hey, saying hi to Sam. The siphonophore human.
It's like the Who's in Whoville. We are here. We are here. We are here.
Ah.
Very cool.
That
Wow,
is really
I didn't even know
that's
they could
neat.
move that fast.
I think it's a little bit of it just being in the current. I mean, looking at how fast the particles are going by.
Mm-hmm.
I don't know if they're actually able
Yeah,
to
they're like-
open and close like that.
They're actually like, this is a good time to feed.
Yeah.
I just know we've tried to do dives in the Anacapa Passage and not looking for this.
That's
Down
not
this
just
deep
the
at all, but
current,
looking-
though. That's, yeah.
Yeah,
It's
it's
definitely
it's
working it.
actively
Yeah.
That's wild.
it's working it tonight. But I know we're looking for white abalone and this Anacapa Passage as well, and we had to wait.
Okay, out.
It's
zoom out.
amazing. The current is really strong and get up to a couple knots pretty quickly, and you have to just time it just right to splash to get your work done.
Is there anything on the list tonight that is a must find? Whoa, hello, crinoids on sponge.
Oh.
Oh wow.
Other than the sponge collections and the usual, no, this does not have any kind of like orange cup coral.
Okay,
situation.
that's exactly what I was going towards.
Oh, I'm so happy to hear that.
No, the priority
Yeah.
tonight, which we may not get to because we still have 2 more watch shifts after this, would be
It's like we're getting up
a
on the
habitat
top of the
mosaic.
hill here. I can see over the top,
Yeah.
though. I don't know. There's Argus is showing up.
What do you think?
That's why we're experiencing the current, right? We're coming up to the top of this area.
We can drop down a little bit.
We're losing our protection of the reef.
Well, I can move ahead
Or
some upslope. So, I mean, I'm kind of falling off, I think.
Yeah. Are you happy with Argus, though, or we want to drop
Yeah,
down a little bit?
I think if I come up slope, then Argus can go up and it'll be okay.
Okay.
We can see over the top.
Yeah. Ship just completed a move too, so we've got a little time to investigate. It's looking a little close in Argus cam.
Yeah, it's still showing 12 meters. Though.
Okay.
What's the preferred positioning of the 2 ROVs when you've got like a ledge like this? Like Argus kind of looking from the front or kind of behind Hercules?
Well, currently Herc's a little bit out in front of Argus. Wait, that's not what's happening here.
No, it's not.
Argus is out in front of Herc.
Oh, hold on. Let's do it. You want to do a reset?
But we're not that off, right? Yeah, we're okay.
Oops. Yeah.
Yeah, I'm behind the times.
Okay.
There.
Lots of urchins as well. I feel like we haven't seen as much up until this point.
Well, and those shells on the rocks are also the brachiopods potentially too, which
Yeah,
seem to
which
be
we haven't seen
pretty
much of.
abundant here.
That looks like a stalked version of the Mycale sponge that we just passed.
Oh yes, so Abby's commenting, seeing lots of little barrels. Okay, here's a scientific name, Rhizanella gaddis. Hopefully I said that okay. And that is the club sponge. And she thinks we just went by one. Or they also could
like
be a stalked Mycale.
So many little of these guys. It's crazy. It's a good spot.
It's not there. That red.
Yeah.
Oh, a little black
Can you
coral.
zoom in now?
Gotta.
Oh, look at that. Nice.
And a passy? And do you know approximately how far it's been since the last watch did a transect?
Let's see here. It's been about 220 meters.
Okay, thank you. Maybe we'll keep an eye out for
All the polyps are out. I mean, I think. I'm sorry. I was just noticing because of the current that it just seems like everything is just working the current to feed right now. It's a very
dinner time.
I mean, just everything seems alive and active right now, taking advantage of the conditions.
Steve, is that a tiny mushroom coral in the
Argus
upper
is
right-hand corner?
pretty dang close to this wall.
Yeah, let's step off